student, and as we can hear from our accent, we are from France, in Bordeaux University and Bioengineering School. We work on elastic proteins that show elastic properties, and we would make a product that would be an alternative to petrochemical plastics and chemical plastics such as bubble wrap and Kevlar. We selected 3 proteins that showed elastic properties. The first one was the spider silk, used by spiders to make webs. The second one was the resilin, that allows insects to fly and jump. And the last one was the elastin that gives elastic properties to skin and muscles. Here is the outlet of our project. We selected consensus sequences from natural protein, and we designed our genes. Then we also designed fusion proteins to test new biomaterials and see if we can have associated them for different qualities application. We cloned the ELP gene we want to clone ELP gene into E. coli to produce and purify our proteins, and then test the mechanical properties. Let me explain how can protein be elastic. We can see repeated sequences look like a spring in natural elastic proteins. And we designed synthetic one with only repeated sequences to have a more modular and reductionist system. So I will let Jim explain you how design our gene.
So the first step to realize our project was to determine the consensus sequence of the resilin, elastin, and silk protein. Here we detailed the silk sequence, but the mechanism is similar for the others. So we identified different repeating patterns. The first is highlighted in blue, and the second in red. And this is how we obtain our protein sequence. This. And we proceeded the same way with the resilin, with this sequence, and for the elastin, with this sequence. But the next step was to design our biobrick. To insert that we could assemble our peptides to compare their characteristics and see how they affect each other when combined. But there is a problem. Indeed, with the current biobrick standard, it will be impossible to produce this fusion, and this because the open welding frame, which is including a stop column. So we modified the biobricks by adding an NHE1 restriction site, and this permits to produce a protein. You can see the compatibility between NHE1 and XBAR1 and SP1, and with this compatibility we can digest by NHE1 instead of SP1. And like that, our production is now possible. And we call this strategy the new border standard for assembly. But it's also important to notice that the parts using our standards could be in any order, so we can create fusions in C terminal and in N terminal. And it's also important to notice that our standard is compatible with every other IGEM standards. So now, we got our custom C sequence and our new biobrick design. So we have to synthesize our seq gene. So we start by designing a double-stranded DNA, and we derive 7 overlapping oligonucleotide fragments. Then, by PCR fusion, we first link in each fragment 2 by 2, and then so on until we obtain our complete gene. And at every step, we have observed the advancement in a gel, and the last step represents a band, which is 350 base pair, which is the expected length to our seq gene. So now we got everything, so it's time to produce.
We started our experiment by using a clone given by our chemist advisor to produce elastine in E. coli. So we needed to work with the proteomic lab because we haven't needed material to realize our production. So imagine students running into 2 different labs in different floors, carrying centrifuge pods and Erwin Mayer downing stairs every day. So it was a really sportive internship for each one of us. So as you can see, we produced 58 liters of production when none of Erwin Mayer was broken. As you can see, having more than 10 students in the lab, it was really, really fun for each one of us. So the critical characteristic of elastine is thermal sensitivity. So the elastine is totally soluble at low temperature and precipitates at high temperature, so which we can visualize by the color change. This reaction and behavior is totally reversible. We can do it again and again. We obtain the same results. The idea is to take advantage of this property to purify our ELP by inverse transition cycling, which is in alternance of hot and cold centrifuge to obtain pure products. So we began with our cell lysate. After warming, the elastine will aggregate, and the hot spin permits the precipitation of the elastine in the pellet. After that, we remove the supernatant and redissolve in cold PBS. So the cold spin permits the precipitation in the pellet. After transferring supernatant, the hot spin will precipitate all the protein in the pellet, and our pellet will be enriched in elastines. After removing the supernatant, we dissolve in cold PBS2, and we obtain elastine enriched supernatant. So the purity of our protein was confirmed by SDS page, and its molecular weight was confirmed by mass spectrometry, which is here. So then we lyophilized our protein. We obtained 87 mg of protein in 3 moles. So this protein was redissolved in water for treating. Then we tried to make fiber of our elastines in our lab. So we tried to make those fiber by injecting solution of concentrated pure ELP in heated salt above the critical temperature sensitivity. And the result is here. So this way doesn't work. After that, we contacted the Center Research of Paul Pascal. So it's funny, they are working on the nanofiber fiber, but we heard only about their work on TV. They are located in the same campus. After that, the lab engineer advised us to use alginate as matrix to form our fiber. So the alginate is polysaccharide extracted from algae. The alginate and mixed with our ELP can form a fiber in a chloride calcium solution. Let you see the result. exaltation of our advisor in front of the accomplishment of 3 months of work. So our tree is totally resistant, so that's why we could do some physical tests. My friends will talk about it.
If you were wondering what our background was, it represents our elastin fiber in electron microscopy. Indeed, alginate and elastin have been compared on microscopy, and we observe a difference between them. But we don't know if this difference is really significant, since we didn't have time to test more samples to check if the result is homogeneous and reproducible. However, we made some other mechanical tests and we applied them into our fiber.
To characterize our material, we went to the Paul Pascal Research Center, which is a chemistry lab across the street from our lab, in the purpose to make some traction tests on our elastins 10% and on the model to compare them alginate. We made the tests on a mechanical test machine which applied a tensile strength on our fiber thanks to a moving crosshead here. In this animation, you can see the you can see the moving crosshead applying a tensile strength to our fiber until it's break. This mechanism allows us to measure the strength applied and the resulting elongation. We extruded our fibers into a calcium chloride solution by manual injection. As you can see on this video, the width of the fiber varied widely. So the results were not reproducible, and this will not be applied for traction tests. So we changed our way to extrude the fibers. The Paul Pascal Research Center allows us to use their patented machine in order to create fibers in an automatic way. So the fiber is extruded into a gradient concentration of calcium chloride, and then it is driven into a bath of water by automatic wheels at a controlled speed. Then it is laid into an oven in the purpose to dry it, and then it is rolled on a spool. Here you can see the results we obtained for a temperature of 25 degrees Celsius. As we created our fibers with an automatic machine, we knew the diameter of the tested fiber, so we were able to produce a stress-strain diagram which gives us the typical properties to characterize our material. If we compare the Young's modulus, which gives us the stiffness, we can see that our Young's modulus is lower than the one of alginate. So it means that our material is more resistant than alginate in its elastic field. The elastic limit is the higher stress the fiber can resist before having permanent deformation. We can see that it is important to say that the elastic field is the domain where the material must remain for the applications. So we can see that our elastin limit is lower than the one of alginate, but it means that our material is more resistant, but only in a shorter field. The maximal tensile strength is the higher stress the fiber can resist before the break, but it's in its plastic field, so okay. The strain energy, if we compare it for alginate and elastins 10%, is the energy required to break the fiber, and we've seen that as the percentage of elongation, which gives us information about ductility, they are almost equal to alginate and to elastin.
That's what we have accomplished. So, we said we have found consensus sequences for elastin, spider silk, and roselin, and we have designed ELP, SLP, and RLP gene. We cloned the ELP gene into E. coli to produce and purify 87 milligrams of pure elastin, and we characterized by mechanical test the fiber. Then we have also produced fusion proteins. No, not produced. We have designed fusion protein and make our new product standard to release.
So perspectives. As first short-time perspectives, it would be interesting to change the ratio between alginate and elastin and see if the critical properties could change. Another perspective was to produce fused protein and with various combinations. Use the better combination to apply this into industry to, as said previously, chemical and physicochemical. And finally, we have shown that the elastin characteristic to precipitate at high temperature, which allow to purificate by ITC really easily, and combine it with our new standard, the both could be used to purify some other protein for the future IGEM teams. Now let's talk about the policy and practice.
So IGEM2014 was truly present on social networks to communicate about its project and its team. We were present on YouTube, Twitter, and Facebook. We also had a lot of reports on our team, such as in blogs of biotechnology, the City Hall of Bordeaux, and the ELCB, where we done all our work. And on the local radio campus, on a great journal, and on regional journal, and on regional TV channel. IGEM Bordeaux was interviewed by two famous media in Bordeaux, France 3 Aquitaine and Sud-Ouest, which made respectively a video report and a newspaper article on our team and our project. Altogether, millions of people were sensibilized by bio-inspired material. A very important part of our work was to find sponsors to finance our project as Bordeaux University has the capacity to finance us entirely. We sent almost 200 emails with our sponsoring document that you can see here, and this allows us to earn over half of our provisional budget, thanks to 12 sponsors. We also use the crowdfunding site to finance our project, thanks to the generosity of our donors. We earned 11% of our initial solicitation.
The 3 months at the ICB was a great experience for each one of us, so thank you for all people who helped us, engineers, researchers, and especially our advisors here, and all the persons who contribute in our project in Indiegogo, and especially our sponsors who permit to us to come in Boston and lead this incredible experience. So we are open for questions. Thank you.
Very nice results. Any questions from the judges?
So you're one of the few teams, and congratulations on that, who actually got to work with your project. That's very impressive. So now you actually work with your product. Can you give us your thoughts about how cost effective would it be to make all these liters and liters of E. coli to get your soap, as opposed to, I don't know, squishing spiders?
Okay. In fact, we work on land layer, and we just use initial bacteria, and we do cell culture into TB medium. So we don't really calculate the cost, but it's not very expensive in labs, and we know that industrials have a cheaper product and can do the production into lab.
Yeah, of course we used medium that is quite costly because it was on a small scale. But if we want to scale up, we will need to improve our process. So to discuss with industrials which are using different processes. Also, we can improve our downstream processes for purification and in order to scale up all of this process. So actually, currently the cost is lower with actual materials, but we do think that we can achieve better costs if we do a proper study on this.
I have a question for you about regulatory systems. Have you delved into, or did you have discussions about how you would get through the regulatory process in your country to begin to use your, produce your product at an industrial scale?
I think there are already some companies that are producing biomaterials with a proper regulatory with not issues at this level. But of course, we would need to prove that there is no risk, no safety issues to produce this material.
Do you know who your regulator would be if you need to contact them to start that process?
No, sorry.
Okay. So I'm the policy and practices judge, or one of them, and so this is something that we're always encouraging teams to think through, because if you've got a really great product, and kudos to your team and all your great work, there are a whole bunch of other things that have to fall into place to actually get the work from the lab into the marketplace. So I would really be interested to know if you could think through some of those questions. And I know also in your country, in your region, there's a fair debate about the use of biotechnology. So I'm also wondering if you had some discussions in your team about the work that you're doing with genetic engineering and how that is perceived within society. Did you have those kinds of conversations?
Yes, sure. We discussed with each other about all these safety issues and also to the ethics of projects in IGEM in general. And we came to also other students and high schoolers to talk about synthetic biology, to make it known in a wide audience in the public, including with our mediatization. So it allows us to have a lot of discussions at this level, and in particular in our projects, that people were quite encouraging about this project, because when we say, okay, it could maybe replace some plastics made with petrol, of course, it's interesting.
Okay, thank you.
I was very impressed with the work, and I would seem to go back to the registry. When I checked the information in the registry, there's a comment that I didn't notice in the wiki, but it seemed to indicate that you were in the ELP work, that you were basically using stuff made from the host plasmids from the host lab that you were simply expressing. Can you comment on that? Is that what you did, or did you make the parts that you were characterizing or expressing?
All right. So we use. So we work on we use the plasmid from our chemist advisor because we wanted to start earlier the project, so we have used our clone for the first failures. But after we've done the cloning into standard plasmid for production, we have done it we have done the production with the standard plasmid.
Any other questions from the judges? Any questions from the audience?